Hi students, welcome to HSC Biology and Module 6, Genetic Change. This is video number 17, and in this one we're going to be looking at cloning, and especially whole organism cloning. The learning intention for this particular video is for you to be able to investigate and assess the effectiveness of cloning, which we're going to be looking at in two different contexts, but in this particular video, focusing on whole organism cloning. To be able to do this, we'd expect you to be able to define the term clone, to be able to contrast gene cloning and whole organism cloning, and then to talk about the effectiveness of whole organism cloning as a reproductive technology, and perhaps to be able to assess how effective it has been. So cloning then is the process of reproducing identical copies of the genetic material, and that's the key phrase here, identical copies. Now, because we're talking about identical copies, we can do the copying process on a very small scale or a very large scale, and it's really scale that is what's going to determine exactly what type of cloning we're looking at. So on the small scale, we're looking at gene cloning. So when we're looking at the gene level, that's something that we talked about previously with things like CRISPR, having a certain proportion or part of a gene that we want to reproduce over and over again. That sort of thing has happened also, for example, with the production of things like human insulin. But that's something that we'll look at in our next video. In this one, we're going to be looking at the individual level, that is whole organism cloning. And that's a much bigger undertaking in the sense that what you're trying to do is to produce an exact copy of an entire individual, not just a particular gene. Reproductive cloning is a method that we use to make a clone or an identical copy of an entire multicellular organism. So that means that we've got to be able to get in early enough to put the genetic information from our clone into that egg before it starts the cell division process. So therefore, every single cell is going to have a copy of the genetic information from the donor parent. So if we kind of try and follow this process down, there's a lot in it, and diagrams are obviously the best way to help you to, particularly if you use something like a flow chart, to identify this. Some of the important things though that you should be aware of, or at least that we can contextualize in the process of cloning, are to review our understanding of sexual reproduction, which is the uniting of two different cells, two haploid cells, an egg and a sperm or gametes, to form a diploid zygote. And that diploid zygote has brought together the two halves, if you like, cells that have been divided into half the genetic material that restores that diploid number. The zygote nucleus contains the important genetic information that's required to produce a new individual, and it has components from the mother and the father as they come together. However, in early embryonic development, there's a requirement that the cytoplasmic material contained within the egg cell is involved or in some way helps to facilitate that early development. And this is kind of one of the areas that we started to realize could actually help to drive this process of cloning. If the haploid nucleus of an egg cell was replaced with a diploid nucleus from a donor, that is any other cell really of the same species, of course, then it could, having still the cytoplasm of the original egg, develop what we'll call normally, but with the genetic material from the donor rather than from the original egg inside of that cell. This sort of transfer is known as somatic cell transfer, and that's we know that somatic cells is a reference to body cells, so non-gametes, non-sex cells. And so the material that's in the nucleus of a somatic cell can then be transferred into that egg cell, and therefore we will have turned a haploid cell into a diploid cell. And if we can ensure that that process of development occurs, then that could then potentially grow up into a complete new but cloned individual. This technique of cloning has been used in what we call therapeutic cloning, which is actually something that's been investigated in terms of human reproductive technologies. There's obviously some very significant ethical issues associated with this, and we will explore these a little bit more in class. But the use of embryonic stem cells can actually be used to sort of help to try and correct or address certain medical conditions. And as I said, there's an ethical argument around that. And it's also been used in reproductive cloning. And probably the best known whole organism clone is Dolly the sheep. And so this technique is a little, I was going to say it's old, it's not that old, but comparatively speaking, it's relatively old. So over 20 years ago now, Dolly was cloned using a cell from a sheep's mammary gland. And so that was the type of somatic cell that was used as the donor. And then that nucleus was then introduced into the egg. The actual original egg's nucleus was destroyed. And so the nucleus from the mammary cell was then inserted into the egg cell. And the cell started to divide. And then once it started to grow, it's basically the process of in vitro fertilization at that stage. We know that the cell is starting to divide, starting to become a little embryo, a ball of cells, and then it was implanted back into the host ewe and developed into Dolly. Now, one of the problems that cloning has, or Dolly certainly opened up, and something that we haven't fully solved yet in this whole area of whole organism cloning, was the fact that there were some issues with Dolly's longevity. And there were question marks, that is, she seemed to age too quickly. She seemed not to be able to have the same sort of lifespan as a sheep that would have been produced naturally. And we think that maybe that might have had something to do with the fact that the cell that was used as the donor cell was from an already adult sheep. And so therefore there was perhaps an issue around the aging. Dolly was aged before her time. And it's created a really interesting area for additional research around this whole area of cloning. I think one of the things that's very important when you're talking about cloning, and certainly when you're talking about some of these important reproductive techniques, is that you create some diagrams to help you explain things. So what you can see is we have two separate individuals. So we have our mother, if you like, call her the birth mother if you want, but we also have our donor. And so the donor is going to be the one that supplies the actual genetic material. So the mammary cells are going to supply the genetic material, which is going to lead to actually down here into Dolly. So that means that all that we're getting from the birth mother in this situation is the cytoplasm, is everything basically within the egg without the nucleus. So we have to enucleate that. We have to remove the nucleus. We then have to add the other nucleus that we want into the cell. Usually there's some sort of a trigger that helps the cell start that process of division. And once it starts that, one cell dividing into two cells into 4, into 8, and so on. And then once we start to see that embryo starting to develop, then that is when it's implanted into our host mother, and she gives birth to the little clone of Dolly. Cloning is a technique that's used more, certainly more commonly in the aspect of gene cloning, and that's something that we will be looking at in the next video. But there's now, from Dolly, there's been quite a number of different animals that have been cloned in a similar kind of a way. So we're understanding a little bit more about this whole process of whole organism cloning in the 25 odd years since Dolly was first cloned. But it's still an area that we need to look at in a little bit more detail, and so we'll do that with a case study in class. Thanks for watching.